Welcome to the video lecture for section 1.3. This is for Statistics 2023. My name is Jorge Gavillan. So in section 1.3, we're going to be talking about collecting sample data. And so the key concept for this section is the method you use to collect sample data influences the quality of the statistical analysis. We talked about this a little bit in the previous videos. So of particular importance is always the simple random sample. If sample data are not collected in an appropriate way, then the data may be so utterly useless that no amount of statistical torturing can salvage them. So the way you collect data is important. Literally, if you collect it in a garbage way, whatever you end up with is just going to be garbage. And so the gold standard is you randomize. And so randomization with placebo treatment groups is sometimes called the gold standard because it's so effective. Now, a placebo is something such as a sugar pill that has no medicinal effect. And so we're not really going to do this in this course, but we are going to talk about random samples. So some of the basics of collecting data. So statistical methods are driven by the data that we collect. So we typically obtain data from two distinct sources. It's either observational studies or experiments. And so an experiment applies some treatment and then proceed to observe its effect on the individuals. And this is usually in a medical study. So individuals in experiments are called experimental units or cases, and they are often called subjects when they are people. Observational study is observing and measuring specific characteristics without attempting to modify the individuals being studied. So experimental, you do something and then you collect the data, and observational, you sort of sit back and collect the data. So here's an example. Observational study, ice cream and drowning. So observe past data to conclude that ice cream causes drownings based on the data showing that increases in ice cream sales are associated with increases in drownings. So the mistake is to miss the lurking variable of temperature and the failure to see that as temperature increases, ice cream sales increase and drownings increase because more people swim. The main variable here would be the temperature. There's also data that shows that ice cream consumption is correlated with murders and is the same situation, just temperature. An experiment. So conduct an experiment with one group treated with ice cream while another group gets no ice cream. We'll see that the rate of drowning victims is about the same in both groups, so ice cream consumption has no effect on drowning. Here, the experiment is clearly better than the observational study. So then we talked about replication. Replication is simply the repetition of an experiment on more than one individual. So good use of replication requires sample sizes that are large enough so that we can see the effects of treatments. The next thing is blinding. Blinding is a technique in which a subject doesn't know whether he or she is receiving a treatment or a placebo. Blinding is a way to get around the placebo effect, which occurs when an untreated subject reports an improvement in symptoms. Another thing is a double blind. Double blind occurs at two levels. The subject doesn't know whether he or she is receiving the treatment or the placebo, and also the experimenter doesn't know whether he or she is administering the treatment or the placebo. Randomization is used when subjects are assigned to different groups through a process of random selection. The logic is to use chance as a way to create two groups that are similar. So perhaps this is the most important thing, a simple random sample. So a sample of n subjects is selected in such a way that every possible sample of the same size n has the same chance of being chosen. And one thing I want to point out here, when we talk about sample size, we use lowercase n. When we speak of the population, we use an uppercase n. So a simple random sample is often called a random sample, but strictly speaking, a random sample has the weaker requirement that all members of the population have the same chance of being selected. So the first type of random sampling is what we call systematic sampling. So systematic sampling, you select as some starting point, and then you select every kth element in the population. So in this case, we have a line of vehicles, so we start with the third vehicle, and that's our starting point, and then we're going to choose every third vehicle, and then so that's systematic sampling. The next one is convenience sampling, and this just basically use data that are very easy to get. For project one that you guys are going to be doing, essentially you're going to be doing convenience sampling. The next one is stratified sampling. So in stratified sampling, we subdivide the population into at least two different subgroups so that subjects within the same subgroup share some of the same characteristics. And then we draw a sample from each of the subgroups. So Stratified sampling, you separate them. Each group have something in common, and then you take a random sample from within that group that you just separated. Cluster sampling is a little different. Here you divide the population area into sections of clusters. Then you randomly select some of the clusters and you choose all of the members from those selected clusters. So that is the key difference between cluster sampling and stratified sampling. Where when I divide them into groups, stratified, I simply randomly sample some of the members of each group. When cluster, I choose the clusters or the groups, if you will, and then everyone within that group is sampled. The other one is multi-stage sampling. So we collect data by using some combination of the basic sampling methods. In a multi-stage sample design, pollsters select a sample in different stages, and then each stage might use a different method of sampling. Observational studies observe and measure, but do not modify. So some types of observational studies: cross-sectional study, data are observed, measured, and collected at one point and not over a period of time. Retrospective or case control study, data collected from past period by going back in time through examination of record, interviews, and so on. And then prospective study, this is just data that's collected in the future from groups sharing some common factors called cohorts. Confounding. Confounding occurs in an experiment when the experimenter is not able to distinguish between the effects of different factors and try to plan the experiment so confounding does not occur. What happens here is that maybe two variables appear to have a relationship, but in fact there's a third variable or a third situation that's causing these two to appear to be related in some way. So controlling the effects of variables. So completely randomized experimental design. Assign subjects to different treatment groups through a process of random selection. And then the other one is randomized block design. A block is a group of subjects that are similar, but blocks differ in ways that might affect the outcome of the experiment. Match pair design. Compare two treatment groups by using subjects matched in pairs that are somehow related or have similar characteristics. Or rigorously controlled design. Carefully assign subjects to different treatment groups so that those given each treatment are similar in ways that are important to the experiment. Now, sampling errors. No matter how well you plan to execute the sample collection process, there is likely to be some error in the results. So sampling error occurs when the sample has been selected with a random method, but there's some discrepancy between the sample result and the true population result, such as error occurs from chance sample fluctuations. Non-sampling error. Non-sampling error is the result of human error, including such factors as wrong data entry, computing errors, questions which bias wording, false data provided by respondents, forming biased conclusions, or applying statistical methods that are appropriate for the circumstances. The next one is non-random sampling error, and this is simply the result of using a sampling method that is not random, such as a convenience sample or voluntary response sample. And that's the end of section 1.3. Thank you.